And Janice.
Afternoon, miss.
Okay, so this was the last question in your genetics worksheet number 2. This one has to do with sex-linked disease, and we all are familiar with once it's a sex-linked disease, we have to use the sex chromosomes in our working. So the first part with part 8 is just to define some terms so you can get out your answers, be ready to answer and partake. All right. So number 1, define the term gene and dominance. All right. So Tristan, I'm selecting you first. Are you all seeing my screen? Are you all seeing the question? Everyone able to see my screen?
Yeah, miss, I'm seeing your screen all that.
Right, gene
Yes,
and
miss.
dominance. All right, anybody else wants to this define what is a gene? How do you define a gene? You'll take really long to answer.
Miss, for some reason the screen is gone.
All right, let me see. In the meantime, someone can at least call out what is a gene, what is dominance.
A gene, miss, is the basic unit of inheritance and is made out of 2 alleles, and dominance is used to describe an allele that is expressed.
Very good. And that's 3 marks that this question carries, so very good. Moving on to the important thing now. So hemophilia, as you know, is a sex-linked disease. Mrs. Trinbage is a carrier for hemophilia. Now, if she's a carrier, it doesn't mean that she has the disease. It just means that she has a recessive allele. So she's not affected. So I basically give you the meaning of the term carrier. A carrier is used to describe the heterozygous dominant form. So when you hear the term heterozygous dominant, you automatically think that they have both the dominant allele, so they will have like capital H, which is normal, and then they will have the recessive allele, which is common H. So they're carrying the recessive trait, but they are not affected by the disease since they have the dominant normal allele that will be expressed. All right, so you should have those in your meaning for carrier. Make sure and
Miss,
mention.
if you could please repeat the meaning for carrier again.
Carrier is used to define a heterozygous. Perhaps I'll type it here. So a carrier is used to define or describe the heterozygous dominant condition This means that an individual has both the dominant and recessive allele. However, only the dominant allele is expressed. Therefore, they will not be affected by the disease. Right. So this is a really nice definition here. All right, so let's go into the working now. So they have a Punnett square already drawn for you. They give you the actual genotype. So they're telling you that the Mrs. Trinbage is. Now, if you can't see it, I'll zoom in a little more. She has the carrier, and he has. How would you describe his genotype? Is he suffering from? Is he does he have hemophilia or is he normal?
He's normal, miss.
He's normal.
He
He's
is
normal.
normal. Yes, exactly. And you all are seeing capital H. Now there's a little error in this paper. What is the error in this paper here? With the symbols.
Miss, it has 2 capital X's for Mrs. Tinbridge.
No, but that's correct. XX is female. XY is male. But there's something about sex-linked diseases. And it's on the notes. Why? Okay, you're seeing that the Y has a capital H. Normally, sex-linked, well, it's not normally. Sex-linked diseases only are carried on the X chromosome. There's nothing on the Y. So when you look at the question above, there was another question with hemophilia disease. Right here. Remember this question? Same thing. But notice on the Y, they put a minus sign. And that is simply to illustrate that there's no allele present on the Y. Only X chromosome will carry the alleles, right? So please remove the H in your working because that was an error. It won't change the answer, but it was an error. Okay, so let's work it out. So we have, remember I told you, put down your sex chromosomes first. Always remember it's female and male. I have seen persons crossing 2 females by accident. All right, so you have female and male. Then you put in your lines. Oh boy. Put in your lines. And then your alleles now. So you have normal, hemophilia, allele, then normal. Right, so now we're going to cross. Right, so you always cross, I would say recommend you cross your sex chromosomes first. So X by X, X by Y, X by X, X by Y. All right, so now we're filling in the alleles. So you're crossing the alleles now. So H, capital H by capital H, you put your 2 capital H's. Capital H going on the X. Nothing goes on the Y. And now we have, okay, in this side, we have big H multiplied by small h. Always put your dominant one first. Right, so that's big H, small h. And then lastly, we have small h by Y. So we put the small h on the X chromosome. So here we are seeing out of these 4 chances, now they say describe the possible phenotype. So you have to list it out. So in words, in writing, you're going to have something like, okay, let me expand a little bit, right? So you're going to have something like, okay, F1 generation genotype. So you put your X, age, well, you listen out everything there. Now for the phenotype, you describe this first female as she's normal. So you put normal and you put female. What about this female here? How are you going to describe her? This one here.
Female carrier of hemophilia.
Female carrier. You don't even have to put all that. Just female carrier. This male down
Okay,
here.
miss. A normal male.
Normal male. And last but not least, the affected person here. They put in hemophiliac. If you're suffering, you call it hemophiliac. That's like the adjective. Hemophiliac male. All right. Now, if you have to put a ratio, out of 4 chances, there's 25% chance, or 1 out of 4, that a child may inherit hemophilia. However, between the 2 chances of the male, because there's 2 chances you're seeing here, 2 chances female, okay? Between these 2, you have 50% chance the male can either be normal and 50% chance the male can be hemophiliac. But between the 2 female chances, you have 50% chance the female will be normal, 50% chance the female will be carrier. So it just depends on how they ask the question. If they ask you what is the ratio or what is the possible probability of a child inheriting hemophilia, well then you say there's a 25% chance the child will become will have hemophilia. If they're asked in specific in terms of between genders, then it will be 50-50. You follow? Luckily, this question didn't ask anything about ratio or anything. They just asked to describe the possible phenotype. So this is the answer here. To get your 5 marks, you need to fill in your boxes correctly. You need to also indicate the F1 you will have a line where you will have a F1 generation genotype. So you just list it out. All right, so there you go. Let's move on to the next worksheet. So we covered this one. Let's go to the second one. The other one is a little more challenging. All right, so we have some sex-linked questions here. It might be a little small on your eyes, so feel free to open it in your window. Let's go to question. Right, I want to tackle the a sex-linked one here. So here we are seeing color blindness, this number 6. Anyone have answers for number 6? This one right here. That looked like a no. Shiva, did you look at last week's recording? Indicate in the chat if you looked at last week's recording, so I know that you're kind of keeping abreast with the classes. All right, since you all, right, now if you look at part of it, these questions will be a little confusing. Huh? So I recommend that you finish it off. All right, so let's move to question 6. Now, the two types of sex-linked diseases that they can bring will be hemophilia and color blindness. All right, now they call it red-green color blindness. That's the name of the disease. It's also a recessive disease, is a sex-linked condition. It is governed by the allele small r. So again, they're always asking you what is meant by the terms. So one, what does recessive mean? I want someone to tell me. Jaden, what is recessive?
Recessive is the allele of a gene that is being masked.
Okay, or not expressed. All right. Ryan, what is sex-linked?
Miss, I don't know the whole definition, but like I know it has something to do with like not determining the gender of the offspring.
Now, I don't want you all to just be memorizing notes because that could conflict with information. When you see a word sex-linked, you cannot get the meaning. So it has to do with an allele that is linked to your sex chromosomes. So anytime you see the word sex-linked, it have to do with some kind of allele that is being attached or linked to the person's chromosomes itself, the sex chromosomes, and in particular the X chromosome. All right, so anytime you see this word, you think of a allele that is being attached to the X chromosome. And that's simply a definition, how you explain the term. Sex inheritance or sex determination has to do with the inheritance of the sex chromosomes, nothing to do with diseases. So you see how if you are just robotic and you're just learning definitions, you're going to mix it up. All right, let's go to B, the working. Use a genetic cross diagram. So this alone telling you how to use a test cross. Right, they specify here. To show the offspring of a normal male and a heterozygous female for this condition. So let's start with the first step. Let's write out our alleles. So we have common R is the color blindness trait. We already established that. All right, so let's put that. So therefore, capital R will mean normal. Right, this is normal. You all agree? You all have to partake, eh?
Yes,
Partake.
miss.
Right, color blindness. Now let's get the parents. Let's get the genotype. So they're telling you, right? So they're giving you, they give you the phenotype. So you put your parental phenotype. So they're telling you is normal male crossing with heterozygous female. What's our next way we can say it? We just learned it. What's the term you just learned for heterozygous dominant?
It's a carrier.
Excellent. Carrier female. So it means that she carrying the recessive allele, but she does not have the disease. So now that we figure out the parents, now we have to figure out the genotype. But we didn't figure out, they gave you that. They always tell you that. You have to come up with the appropriate symbols. So normal male will be X. Oh, I have to, I can't type those little fonts, so I'll have to draw it out here. Right, so we already established this part, right? Now we have to get the genotype. So normal male is X. So first you put your sex chromosome, XY. female is XX. We don't know what's sex chromosomes. Right, you tell me what is the allele for normal male now? What we put in? Let me see how good Ollie is.
Miss, for the male, they'll put a big R over the X.
Very good. So nothing going on the Y. Eh? The minute you put something on the Y, you don't understand what is a sex-linked disease. Only the X chromosome will carry an allele. Right. Tell me, what is the carrier female now?
A capital R on one X and a common R on the other X.
Very good. So we have capital R, common R. So we have our carrier female, we have our normal male. So they want to know, so you have to use a test cross. So anytime you're doing test cross, put in gametes, put the word gametes. Gametes. That's a line you should have in your working, because this question is 6 marks. Right, and then you circle everything now. It's just a symbol. So you circle your gametes. Right, so now we have to cross. So we multiplying and writing as you go. So X by X, X, X. Big R by big R. So you put it, all right, so I'll put the alleles after. As a matter of fact, I'm gonna do it one time. It gets very cumbersome. So then you multiply X now by the next X, X, X. Then you have big R, big R, and then you have small r. Right? Make sure you're seeing that. Then you come across here, Y and X. Now it has some children, when in exam mode and they're under that pressure, they will put the Y in front of the X. And that is not how you read a sex chromosome. Always X in front of the Y. So we have big R, put it on the X, and then we multiply the last one. X, Y, you put your small r on the X. Okay, you can't really see that. Small r going here. Right, everyone seeing it now? Put down your working as you go, eh? As I'm writing, you write. Okay, so you have your test cross. Now from the genetic cross, state the ratio of normal children to colorblind children. So I want you now to tell me what is the ratio. Colorblind to normal. If you know the ratio, please say it.
Miss, a 1-to-3 ratio.
1 to 3. Now, normally in our ratio, you always put the larger number first. Now they say normal, and you have to follow this. Normal to colorblind. So let's look at normal. Isn't this female here normal? We have normal female. What about this female here? She's a carrier, but she's still deemed as normal. She's not colorblind at all. And this male, he's normal. So you have 1, 2, 3 normal, 2, 1 colorblind male. Well, 1 colorblind child. Never put 1 to 3. You'll get it wrong. Because you are not following this scheme. They say normal to colorblind. That will follow the scheme. All right, so the next part of the question, state the percentage of male children who could be colorblind. So it's now you have to look at these two and tell me. So these two, what is the percentage between the two male as to who could be colorblind? Tristan, what is the percentage?
Miss, like 33%.
Look at the question. They say state the percentage of male children who could be colorblind. Isn't these two the male?
Oh, yes, miss.
So you're looking at these two and you're looking at the percentage between the two male now. One is normal, one is colorblind.
Well,
So
then
what
50%
percent?
chance.
50, right. So you see little maths, maths.
Mhm.
50%. And if they ask you what percentage the female is a carrier, 50% because it's one chance normal, one chance is a carrier. 1 divided by 2 multiplied by 100 give you 50%. Simple maths. Okay. Let's go to our next one. All of these we did this tongue rolling one already. Let us do a codominant question. So I'm going to give you some time. Have we done question 3 with sickle cell anemia? Okay, I'm giving you 5 minutes to work on question 3. All right, and I want you all to take a picture. When you're finished, send it to me directly on WhatsApp, please. Thank you. Okay, so far I get one working. I'm waiting for the rest, huh? So I'm waiting for Tristan, Ryan, Keenan working, Janice, Jaden, Shivan. Send me a picture, please. Then I'll correct. Okay. Now I gave more than enough time to complete this question. And so far I only got two responses, which is very, I'm not sure if you're not understanding or if it's still working, because I didn't get any feedback. So what is it? Is it that you're having
Yes.
problems?
Miss, I'm trying to find the last question.
Number 3, it's on the screen.
No, I mean the
Part
part
C.
C. Yeah, miss.
Oh, okay. Sorry. It's also in the classroom, by the way.
Okay, miss.
All right, so I'm going to start correcting this one. Sickle cell anemia is caused by a change in a gene which controls hemoglobin production. So they're giving you capital N is the unaffected allele. Capital S is the change allele of the hemoglobin gene. So they're trying to say this is the sickle cell allele. Now, if the genotype is SS, the person has sickle cell anemia. If the genotype is NN, the person has normal hemoglobin. Now, sickle cell anemia is not sex-linked. So you're not going to put X, 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 Y. If you started that, you're already getting it wrong. They gave you the actual, no, capital H stands for hemoglobin. Hemoglobin, it's a protein where this allele is found on. So it's just like a symbol, right? So that is what you have to use. So here we go. If the genotype is NS, what is the condition known as? Janice, what you had?
Miss, pardon me. What did you say?
Number 3A, part 1. What was your answer there?
Oh, a carrier.
Carrier. So you call it a carrier. That's the condition because they have dominant and they have recessive. Part 2. Identify one advantage of having this condition. Right. So what you all came up with? Now, this is what you call sickle
Yes.
cell.
I came up with one advantage is a higher chance of surviving malaria fatality.
Yes. Now, in places where there's a lot of malaria cases, so that would be like Africa, in the Caribbean, in the tropics, they tend to have a lot of those mosquitoes. Now, if it is, you have sickle cell anemia, if you are carrier of it, at least carrier, let me just get a sickle cell anemia, just a little picture. Right. Right. So basically, the person they have sickle cell, red blood cells, and they also have normal red blood cells. So if they are a carrier, it's an advantage for them because malaria could only attack normal red blood cells. It cannot attack the sickle cell shape. So that is why it's an advantage to have this condition. Even sickle cell people too. If you have the disease full blown, there's no way you can get malaria. All right, because the malaria disease or the pathogen that causes malaria only attacks the normal red blood cell. So that's the advantage. Right, so let's go to the diagram. So we have to show the production of children by a man. They're giving you the genotype for the man, and then they say a woman with normal hemoglobin. All right, so what you all cross?
What we got, miss?
Sorry, what you cross? What was the genotype you cross?
The normal the normal hemoglobin.
You can just pull
Um,
out the allele.
Okay, so H N H S crossing which HN and HN.
Right, normal female, right? So now if it's something for 6 marks, you have to put out your lines, parental phenotype. So the man, he's NS, so therefore he is a carrier. The woman now, she's normal hemoglobin. And then you put your next line, parental genotype. So you fill in the genotype with the symbols here. All right, so I'll have to write this in. So we have carrier, he is at H normal, H again, he carrying sickle cell allele, but he can still have a normal healthy lifestyle, because he still have normal red blood cells. And then you have the female, she is normal, so she have N and she have N again. Right, so now we have our Punnett square to draw. Oh my, why this happen, right? No, we are not concerned with where the male is placed, where the female is placed. We're not concerned with that. So we have, all right, put down your H's please. I suggest put down all your H's. So that won't confuse you. All right, now allele time. So we have 2 big N. Then we have again 2 big N. Then we have a carrier. And then we have an X carrier. So here we have a two chances of being normal, 2 chances of child being carrier, which is a 1-to-1. So here they are saying from the cross, what proportion the offspring is homozygous dominant. Let's see. Homozygous dominant. Take a proportion. You could represent proportion as a percentage or a fraction. So part 1, you're going to put, right? What percent? Tell me. Anybody there?
Miss 50.
Okay, so we're hearing 50% homozygous dominant, correct? Or if you want to represent it as a fraction, half. Then they say homozygous recessive. So anybody homozygous recessive here?
No.
So you put 0%. Last one now. Heterozygous. Anybody heterozygous? Yes. We also have these two, which encompass 50% as well. You're seeing that, right? Okay, so let's move on to C. Part 1. Explain why it is important for the man and the woman in a genetic cross to produce any children with sickle cell anemia. So basically, it's impossible because both parties, male and female, they all possess the normal hemoglobin allele in the dominant form, or there is no sickle cell alleles that are present in both parties. In order for a child to have sickle cell, it should have been either the female having the disease or the male carrying the disease and the female is normal. So so it's impossible for the man and the woman to produce any children because neither parties had sickle cell that were either in a dominant form or both parents carrying the trait. You all got that?
Yes, Miss.
Last one. Suggest a combination of genotypes that will produce at least one child with sickle cell. So like I said, it's either you have two parents carrying the trait. So let's say you had, let's say it was both carriers. So you had NS here, mating with NS. You will have a child who will have the disease. Or you could possibly have one parent having the disease full blown and the other being a carrier. Any other types, any other cases you think will have a child that has sickle cell? No. If we have normal here with a sickle cell, all children will possess carrier. So this is the only two combinations here. Right? You can always work it out just to make sure. Okay, so that's basically now this is what they call codominance. Sickle cell anemia is a codominant case where both alleles are dominant. Right? So you're only using capital letters. Blood group is a codominant as well. And the two sex-linked diseases that they will bring will be color blindness and the one with hemophilia. Okay, so be mindful of those questions. Let's pause for a break. When we return, there's a really nice topic that you all have, which is diseases. Diseases is a area where you can earn marks because it's a lot of reading and it's not hard to understand. All right, so let's come back at 2:20 and we will cover diseases today. And I want to introduce you all to the past paper questions for homework. All right, so see you all in next 15 minutes.